Gene expression. Firstly, DNA is stored within the nucleus of a cell. Every strand of DNA is made up of repeating units called nucleotides. They are made up of phosphate, deoxyribose sugar, and a base. Nucleotides bond with each other at the 3' and 5' carbon. If we look at a nucleotide at a molecular level, we can see why. There is a carbon chain of 5 carbons within the nucleotide, and the bonds are on the 3rd and 5th carbon, hence the terms 3' and 5'. When the nucleotides join together in a long chain, it's called a polynucleotide. Poly means many, and within that chain there is a phosphate sugar backbone, named as such because the bonds are between the phosphate and sugar. At the bottom of every strand is a free 3' and at the top a free 5'. The strands are held together by the bases. There are 4 different ones: adenine, thymine, guanine, and cytosine, which means there are 4 types of nucleotide. The bases are complementary, so A goes with T and C with G. It's impossible for them to interact with others. A double helix chain is antiparallel because one of the chains of nucleotides goes from 5' to 3' and the other 3' to 5'. Due to the fact that the 2 strands are the opposite way round, they have to be replicated differently. The part of gene expression that occurs in the nucleus is called transcription. First, the DNA is unwound by the enzyme RNA polymerase, and the hydrogen bonds between the bases break to form 2 template strands. Then, for the leading strand, the strand which starts at 3' and ends at 5', a DNA primer, which is a short strand of nucleotides, binds to the 3' end of the template. An enzyme called DNA polymerase then adds free nucleotides in one direction, and this continues until the leading strand is copied and forms the primary mRNA transcript. However, for the lagging strand, the strand which starts at 5' and ends at 3', due to the fact that the DNA polymerase can only add to the 3' end of the primer, it's a different process. Many primers attach along the strand. The DNA polymerase then extends the primers from the 3' end. The enzyme ligase joins the fragments. This is a discontinued process but still creates the primary mRNA transcript. Once this is done, one more step must be taken before the mRNA leaves the nucleus. Not all the parts in a gene, which is a section of DNA, is needed to create the final protein. The non-coding or unneeded regions in the gene are called introns, and the coding or needed regions are called exons. The introns are removed from the primary transcript, and the remaining exons are spliced together to form a continuous sequence. The order of exons does not change during the splicing. This has created what we call the mature transcript. The mature transcript then leaves the nucleus to travel to the cytoplasm. The next part of the process is called translation, and for this to occur, the mature mRNA transcript has to go to a ribosome within the cytoplasm. This is where protein synthesis will occur. Transfer RNA, which is found in the cytoplasm, enters the ribosome and the process of translation begins. A codon is a triplet of 3 bases on the mRNA which carries the code for an amino acid, whereas an anticodon is an exposed triplet of bases on a tRNA molecule. Each tRNA folds to form an anticodon site at one end and an attachment site for a specific amino acid at the other end. Each tRNA molecule carries its own specific amino acid from the cytoplasm to the ribosome. Translation begins at the start codon and ends at the end codon. Anticodons bond to codons by complementary base pairing, translating the genetic code into a series of amino acids. Peptide bonds join the amino acids together. Each tRNA then leaves the ribosome as the polypeptide is formed. Polypeptide chains fold to form the 3-dimensional shape of a protein, held together by hydrogen bonds and other interactions between individual amino acids. Proteins have a large variety of shapes which determine their function. And that concludes how DNA travels from the nucleus to the ribosome and becomes a protein.